Okay. Uh, my presentation is based on the model for songbirds, using songbirds for understanding learning and linguistic disabilities in autism. So this presentation is on how current scientists are using songbirds as a model for understanding the complexities of language to best understand the linguistic disabilities in autism. The word autism is derived from the Greek word autos, which means self. People with ASD, autism spectrum disorder, are often characterized as self-absorbed and seem to exist in their own private worlds. They have limited abilities to successfully communicate uh, and interact with others. This is because they have difficulty developing language skills and understanding what others communicate to them. This is found most in nonverbal communication, such as like hand gestures, eye contact, body language, social cues, and facial expressions. The studies to be presented will mostly focus on the verbal speech and social cues that the songbirds of the songbirds and how they can relate to humans with ASD. The common speech issues are often um, uneven language, skill development, and repetitive and rigid language. This often means saying words that don't have any contextual place in the conversation, repetition of words or syllables without specific reason, and also the omission of syllables within words or phrases. So scientists in the 1970s knew that ASD was linked to genetics when it was first studied on twins with ASD, and they found that it was highly heritable um, through, though they found that genetics did not account for all of the causes of ASD. Some environmental factors, such as maternal immune response or complications during birth, may also work alongside genetics to produce and intensify the features of ASD, though ASD is not found to be caused through um, though ASD is found to be caused through genetics, there isn't any singular ASD gene. However, it's continuously being tallied uh, that more than 65 genes are considered to be to have strong ties to ASD, and possibly over 200 others that may have weaker ties. Less than 1% of people with ASD have it because of any one singular major genetic change. And in fact, females seem to have more genetic mutations than males. This is suggested to occur because of the interaction with the X chromosome, um, where females may have a mutated allele on a single X chromosome, while the other X chromosome contains a wild type, and it leads to an unaffected female. So that requires a lot more mutations to produce ASD in a female. Most research rats. So So most research attempts to aim at the single gene male cases, more often because they are the easiest to create models and experiments for. Often, mouse models are produced for testing the physical disabilities of ASD. Within the last 20 years, songbirds have been the new model used for testing, but for the linguistic aspects of this disorder. Birds have been used for research testing for many decades. They have also been subject to extensive artificial genetic engineering um, for agricultural purposes by large poultry companies, which has led to many great techniques that aid scientific research as well. The three most common songbirds are um, sparrows, canaries, and zebra finches. Um, So in this topic of research, the zebra finch is the go-to songbird model because of the linguistic characteristics the males require to court a mate. By 3 months of age, young males are already have already mastered their distinctive courtship song. They require listening and imitating their father's song during critical periods of development to construct their own song, much like how humans require listening to others to develop language skills, and also like human language, a zebra finch's song is complex and has an important social purpose. Another ideal parallel between songbirds and humans is that the neural circuitry of communication is similar across the two species, involving a loop between the cerebral cortex, basal ganglia, and thalamus. This is what allows the several cognitive processes to be involved in song learning. Juvenile birds must use auditory learning um, to hear and memorize adult song patterns, then later take those learned patterns and practice them until they can reproduce them. This requires auditory feedback and auditory motor integration. After reaching adulthood, the zebrafinch's song is crystallized, and they can no longer learn a new song. This may relate somewhat to humans when they struggle to learn a new language in adulthood. New phonemes um, and sound variations become very difficult to master later on in life. So, a recent study has found songbirds have much greater similarity in neural circuitry and language than originally thought. In songbirds that are learning their song, there's continual integration between secondary auditory regions and sensory motor regions, similar to the integration between Wernicke and Broca's area in human infants, requiring speech and language. Um, Taken together, song learning in birds and speech acquisition in humans may provide useful insights into the evolution and mechanisms of auditory vocal learning. When producing the perfect specimen for each research study, the advances in genetic alterations have been a vital tool. The egg is simple to manipulate and to acquire the necessary specimen, since the egg is easy for a mother to produce. It doesn't require a live parent to incubate, and the egg can also survive without a perfectly intact shell, if well attended by the researchers. Not often, but occasionally, researchers can luck out and find commercially available antibodies that bind to receptors to be able to produce a specimen, even without genetic alterations. So, many recent studies have found various things that have the potential to provide insight in the causes of ASD. Transcription factors and genes, such as the FOXP2 and FOXP1, which is short for Foxhead Box 2, P2. If you recall from the textbook, is a transcription factor likely affecting a large variety of genes. Mutations lead to long-lasting difficulties uh, learning to speak, understanding verb tenses, and having an underactivated Broca's area. In birds, it disrupts their ability to learn songs, and FOXP1 could also be related to many speech disorders. RNA, such as microRNA 9, is an evolutionary conserved microRNA that is abundantly expressed in area X of basal ganglia nucleus, required for vocal learning in songbirds. And overexpression of this RNA results in a song with syllable omission and alters acoustic features. Often, this is found to occur alongside FOXP1, FOXP2 downregulation and disrupted dopamine signals. This dysregulation suggests that in humans it could also contribute to language impairments and may provide the answer to why people with ASD may also omit their syllables. So, hormones and receptor antagonists, AVT, which is arginine vasotocin. So, human and MC, which is Manning compound. Um, Humans and birds are both naturally inclined to motivated to behave socially and mate and survive. Arginine vasotocin, a non-peptide hormone, promotes both song and social motivation. However, the Manning compound is an antagonist, and increases in this causes severe disruption in arginine vasotocin. Uh, Disruption leads to lack of interest to learn songs and lack of interest to sing to attract a mate. Dysregulations in either one of these could be the cause of the self-absorption found in humans with ASD. This lack of interest, especially during critical language periods, could explain this common behavior. Also, other proteins in genes like CNTNAP2, which is contactin associated protein like 2, is a gene that is membrane-bound protein, which is found predominantly in the male's robust nucleus of the archopaleum, RA for short, a cortical song control region that's analogous to the human layer 5 primary motor cortex. Because of this protein, males experience unique neural connections not found in humans when it comes to speech production. So further testing is required to see how this necessarily relates to humans, but it could pose some interesting connections. And for future studies, there's a current study going on now that glial cell proteins caused by gut biome stimuli could be affecting the brain during pregnancy. So ADNP, activity-dependent neuroprotective protein, was originally discovered as a protein that is secreted from glial cells in response to vasoactive intestinal protein, VIP for short. This is a major neuroprotective peptide in the CNS and PNS. A activity-dependent neuroprotective proteins is necessary in the orderly degradation and recycling of cellular components. It is also crucial for brain development and important in brain plasticity, cognitive, and social functioning, characteristics that are all impaired in autism. The interesting fact of this protein is that it is released in response to one's natural gut biome. The most recent follow-up to this study was just released in February, and it confirms that male mice and birds show more of this of issues with this than females, which is in line to how autism affects male humans, and this could lead to promising results in discovery for ASD causes. Perhaps the gut biome shared from the mother and the food that the mother eats during pregnancy could have a potential of affecting the neuroprotective proteins in the fetus, and the potential for harmful or most beneficial prenatal diets could be discovered. This is one of the ideas that I found the most interesting because of how important our symbiotic relationships can truly be, and what benefits could there be if we were to learn to mutually maximize these relationships. And as well, I think of that is, what about our parasitic relationships? So any virus or bacterial attacks can cause antibodies that are targeting the receptors for these parasites. However, we could be degrading our child's receptors as well, causing issues. So I think future studies should be done on that as well. These here are my resources, and thank you. Please comment any questions.